Hello everybody, my name is Babu Bassa and I am an adjunct professor at the Department of Environmental Toxicology, Southern University, Baton Rouge. In today's video, I will demonstrate to you one of my software tools that performs most of the routine computations in a biochemistry laboratory. I will first show you how to download the program from the GitHub site. The program requires Windows 10 or higher operating system on a PC. So this is the web address. You can double click on this to go to the website, but otherwise you can also copy and paste it in the address bar of your browser. Paste and then highlight this address and click enter from the keyboard. This is the site. Once we are here, I encourage you to read this readme file. It has a lot of information in it. Otherwise, go to releases for downloading the program. Click on releases, assets, and this is the file, installer file. Click on that. Once it has downloaded the program to your computer's downloads folder, Windows downloads folder. You can go there and open it, but I can also open it from here, from this menu. Open, run, next. The screen darkens and asks you if you allow the program to make changes to your computer. I presume you want to download. You have no objection to install the program, so you have to say yes on the screen. Yes. Close. The program has already been installed, and you can go to the program menu of the Windows and find it in the alphabetical order. The name of the program is Prototype-A-1. So let us go here. This is the program. If you click on it once, it will open the program. This is the program and user interface of the program. If you want to uninstall, go back to the same shortcut and right click on the shortcut and uninstall. This is the disclaimer because the program gives only instructions, but the careful handling of the reagents is the responsibility of the experimenter. If I say OK, and this message tells the user that the program will be functional only till December 31st, 2022. That is the end of this year. After that, you can still view the documents that you save, but it will not function. And before that, I am going to bring out a professional version of the same program. I will go over each one of the options. The first button, these are all toggle buttons. Toggle buttons means they go on and off if on successive clicks. So instructions, these are general instructions, and please read them. They are very useful for using this program. And then this is a list of 20 examples. When you choose any one of the examples from this drop down menu, the program will make the entries, and then you have to click OK or enter. Then it will display the instructions. I have made this to show you the scope of the program, which is very large. Then prepare solution this is for the preparation of molar solutions. Okay, say anything, some compound. I will say glucose. When you click on these buttons, it will open up fields. These are the fields that have to be filled out before clicking enter and see the instructions here. So I would say glucose content, the concentration of what molar concentration of glucose you want to prepare, say 10 millimole. You have to always select the units from this pad. When you enter the numbers, only the relevant units blink, relevant to this field will blink. Then the volume required, say you want to prepare 100 ml, and then molecular weight, I think it is 180 approximately, whatever. This is an example only, and if you click, you will see the output here. The output consists of all this information, that is for the sake of records, and then the calculation. It displays the calculation also. If you look at it, when you prepare a molar solution, what you need to know is the weight that you need to weigh out. So weight of glucose here, that is 180 mg. Verify the calculation, then the suggested procedure is dissolve 0.18 grams, that is 180 mg solute, and approximately 70 ml solvent. The solvent normally here is water, and then adjust the volume to 100 ml, mix well again. I will move on to the next one. Reconstitution of solutes. We do this very often, because not very often, but sometimes in the biochemistry laboratory. For example, if you are using growth factors, hormones, they come in very extremely low quantities, like 100 microgram, and they are lyophilized solutes, normally sticking to the walls of the vial. So there is no practical way to weigh them out. Basically what you do is take the weight from the vial and then reconstitute it. This option comes in very handy. Although I am giving this growth factors example, growth factors and hormones, it can be used for anything. I will give you an example, like name of the compound. I will put EGF something. Concentration required maybe 10 micromolar, and then weight of the solid, maybe you have 100 micrograms, and these units are very flexible. Then molecular weight, I don't remember, probably might be 2000. Let us put 2000. This is only an example. So enter. You will get the instructions here. I will move on to the next choice, that is the big one, prepare buffers. So I made about 20 buffer calculators. These are all custom calculators for each buffer. So, and this program provides, depending on your input, it can be 3 methods, 1 to 3 methods. And the minimum, first you have to select a buffer, for example a state buffer. The minimum entries required here, the minimum number of fields that you need to fill out are 3. One is the pH. I will say 5, and molarity, I will say 100 millimolar. I want to make a buffer of 100 millimolar, and volume 100 ml. This is an example. And if you have stock solutions of the salt and acid, you can enter here and it will show you one more method of preparing this buffer. Say I will say 100 millimolar, I will say 200 millimolar salt, in this case sodium acetate, salt stock, and this can be acetic acid, 200 millimolar. Then they have to be close to 100 millimolar or more, because you are going to draw solutions from these two and prepare the buffer. That is one way of preparing the buffer. So enter. I do not know why it is highlighting it, but otherwise this is the one. Let me go here. This is the output. As usual, like before, it will repeat the entries here for the sake of records. And then you know that Henderson-Hasselbalch's calculation is everything for the preparation of buffer. Although we do not do that, we do not use that calculation a lot of times. That is the ultimate. So I developed an algorithm wherein the output contains an actual display of the Henderson-Hasselbalch's calculation. Here, you know, pH equals pKa plus log salt by acid, that is the equation. And in your case, you asked for a pH of 5, and pKa is the pKa that comes with the program, and it is the pKa at 25 degrees setting, pKa for acetic acid, and then log salt by acid. So this is the calculation, actual calculation. What we find out from this equation, using this equation, is the actual moles of the salt and acid. You know, a buffer is a mixture of weak acid and its salt, and then you have to mix these two in this ratio. It is not the ratio is here, it is actual moles of salt, in this case sodium acetate, and acid, acetic acid. And then we can start with 0.1 molar acetic acid and adjust the pH. Adjust the pH with sodium hydroxide. That is one way of doing it. The other way is to dissolve these two in this ratio. So basically you will actually weigh out the anhydrous sodium acetate and acetic acid. Actually you can weigh out it. It tells you here. You can read these instructions, and always adjust the pH with a pH meter. The third method is, because you have provided 200 millimolar stocks in each case, you can mix those stock solutions in this ratio, in these volumes. Always please, you have to verify pH with a pH meter. That is important. The most common way of preparing buffers in a laboratory is starting with the acid and adjusting the pH with sodium hydroxide. I will show you here one more buffer, say MOPS, and pH I can put 7. Okay, if you enter a pH that is far removed from the pKa, then it will warn you that the buffer may not be a stronger one. But let me first show you this preparation of MOPS buffer. pH 7, molarity, say again 200 millimolar or whatever, millimolar. Watch out here that these blinking keys, they are only relevant to this field. Volume, I will say 200 ml, anything or something, and then I don't have the stock solutions. Then enter to save time here in the presentation. So let us see what it is describing. Two methods. One method is basically to start with the acid and adjust the pH, and then the other method is mixing the salt and the acid in particular quantities. Another buffer I will show you, any other buffer, say citrate buffer, and if you want to verify the calculation, it shows the calculation, you can verify it, go through this and this. Another way is, for example, pH 4.76, that is same as the pKa, and then molarity 1 molar, and then volume required 1 liter, let us say 1 liter. So by definition, The pKa is pH at half dissociation of the acid. So you should get the results as exactly 0.5 moles required are 0.5. That is half dissociation. Deliberately I have chosen the pKa as pH. That is about the buffers. Please explore further all the buffers under this prepare buffer option. I will next cover the menu item, dilute solution. This is an important one because we do dilutions more often than anything else in a biochemistry laboratory. There are many different types of unit expressions for the concentration of the solutions, and I try to cover most of them in this program. For example, if we have a compound like LPA, which is lysophosphatidic acid, and we have a stock solution of 100 mg per ml, and we want to test at a final concentration of 1, 2, 3, 4, 5, 6. This is an example, mg per ml. And for example, on a cell culture, the cells are grown in 30 mm dishes, and the volume of the cell culture medium is 2 ml. Then if we enter, these are the proportions in which we have to deliver the stock solution to the culture medium to achieve the final concentrations requested here. Normally these compounds are active at very low concentrations, so mg per ml may not be the right concentration. So let us change it, try microgram per ml and enter. What happens is, because of the difference, because the final concentrations are very low, the volume that we have to deliver from the stock solution is 0.12 microliters, which is not really measurable. So we have to dilute the stock solution in order to increase this volume, raise this volume. Sometimes, depending on the compound and its activity, biological activity, those dilutions can be very high, for example, million to billion times. So it is not feasible to dilute the stock solution in a single step, because if we do that, we have to handle liters and liters of solvent. So we have to split the dilutions into multiple steps. For example, we can achieve a million times dilution by splitting the dilution into 3 steps of 100 times each. These multi-step dilutions are called serial dilutions, and so far the serial dilutions are designed intuitively, case by case, depending on the scenario. With that approach, it is very difficult to program, because we have to generate millions of lines of code, and also even after that the programming will be restricted to special scenarios. In connection with the programming of these serial dilutions, I modified the basic dilution equation to adapt it for serial dilution programming. My new equation basically incorporates the volume that is delivered to the culture dish as one of the variables. I have published it as a preprint in ChemArchives. I have given the reference below this video, the DOI. I encourage you to review that preprint, which is not yet peer reviewed. When the volume is so low, my program will ask you to enter a volume that you desire to deliver to the culture dish. In this case, for example, I say 5 microliters. This volume is kept low because these compounds are thermally dissolved in organic solvents like DMSO, which can be toxic to the cells. For example, if you are testing it on a cell culture. So let me say microliters and enter, and these are the instructions, new instructions generated. based on our request of the final concentrations which are low. Although we use mg per ml units when the compound is not really pure, the most ideal units are molar units. So let me change this to micromolar, say micromolar, and see, let us see what happens. Enter. Since these two concentrations are expressed, the start and final concentrations are expressed in two different units, the program will need the molecular weight to convert them into both of them into molar units. So let me say 444 approximately. Enter. Again, the volume that we have to draw from the stock is very low, 0.05 microliters. Therefore, the program will ask us to specify a volume that we wish to deliver to the culture dish. I will set 10 microliters for example this time, and enter. Again, these are the new dilutions, new instructions generated for these specifications. Next, I will go to another unit type that we use more frequently in a biochemistry laboratory. As an example, I will take the titration of serum for antibodies. Let me say serum, and we want to titrate it at different concentrations, different dilutions. So for the stock concentration, in this case, say 1 anything, that is the more appropriate units here. And then we want to test it at, for example, 300, 400, 500, etc. And let us say enter. In this case, okay. Okay, here I have not selected the units. 300, 400 for example, and then units. In this case, the appropriate units are times dilution, fold or times dilution. So select times, and then say final volume 1 ml. We are performing the titration in 1 ml. Say for example enzyme immunoassay or radioimmunoassay. Say 1 milliliter and then enter. These are the instructions generated. Again, here we do not have to dilute the serum to increase the volume. The volume is pipetable 3.3. Anything above 2 microliters, the program considers as pipetable volume, because we have this 2 to 10 microliter append of pipettes available normally in most labs. Let me change this to much higher dilutions, like 1000 to 3000, etc. Then again we have to select times as units and enter. Again, the volume is too low, 1 microliter, which is less than 2 microliters. The program will ask us to enter a specific volume, specify the volume. I will say 10 microliters. We can go all the way up to 1 ml, 10 microliters, which is a convenient volume to handle. Enter. These are the instructions generated by the program. You may please review them. And it also again uses dilution factor to test the accuracy of the dilutions. Dilution factor expected is basically stock concentration divided by the final concentration. Dilution factor achieved is based on the volumes that we mix, and it calculates progressively. At this juncture, I will also show you one more type of dilution that is frequently used in microbial work. For example, if we want to assay a bacterial sample and growing it on agar plates and colony counting. So again, the stock concentration is like 1 anything, and the final concentrations can be 100 times, 200, 300, etc. times. Final volume, specify it as 10 microliters, because these are, the bacterial sample is inoculated spread on an agar plate and grown on an agar plate. There 10 microliter volume is convenient to spread it on an agar plate, solid agar, and there is no real final volume. You say 10 microliters here, for example, you will understand what I am talking about, then enter. Then pipetted volume also, please request for 10 microliters and let us see what happens. Okay, this message says that the final volume and the pipetted volume are same. That is okay. Enter. These are the instructions, and this is very useful when bacterial samples are analyzed by colony counting. I will go next to the other unit types, like next one, volume by volume percent. We have methanol, and then we have a stock methanol solution of 80%. Volume by volume percent, this is a solvent, and then we want to make a 25% methanol out of it. 25%, again select volume by volume, and the final volume, for example, we want to make 200 ml and enter. Then this is we have to mix the stock, 80% stock, in these proportions. 62.5 ml of the 80% stock and 137.5 ml of water, normally water. The next unit type I am going to cover is, next unit type is weight by volume percent. For example, if we have a 5% solution of glucose, select weight by volume percent. In chemistry, percent solutions are defined as grams per deciliter, grams per 100 ml. So if we have a 5% stock solution of glucose and we want to prepare, say 1.25%, again select weight by volume percent, and we want to make maybe 25 ml of it. You can choose anything here and enter. So these are the instructions for weight by volume. We have to mix 6.25 ml of the original stock with 18.75 ml of solvent or SA buffer for 1.25 weight by volume percent of the final solution. Like this, I covered also, if you see this parts per million and CPM, and if you are diluting the cells also, you can use it. Here it is basically, it has only a label value. When you use ppm, the solvent must be water. So that is about the dilutions. Actually you can explore them using the examples also. Thank you. So the next item is write protocol, and I divided the protocol types into 3, and the first one is cell culture testing, titration of serum, microbial samples, cultures, etc. In all these cases, preparation of doses of the test compound is part and parcel of the protocol writing. So here I will say lyso-PC, for example, and then if you have a stock concentration of 10 mg per ml, and then we can request up to 6 concentrations, final concentrations. So 1, 2, 3, 4, 5, 6, I would say micromolar. Where is it? Micromolar. And then volume 2 ml. If you have cultured the cells in 30 mm dishes, that is where we normally put 2 ml. And the intervals like 1 and 2 hours, and we are running the experiment in triplicates. Enter. Verify entries. Okay, here we have to choose 2 hours. Where is it? and then enter. Because these concentrations are expressed in different units, we have to provide the program with a molecular weight. That I will say 440, enter. Pipetted volume. Since the volume is very low, 0.528 microliters, we have to deliver for the 6 micromolar. So we have to specify a volume. I will say 5 microliters. Normally this is dissolved in DMSO, so we will keep it low. 5 microliters and then enter. So this is the protocol. This is how the doses are made. If we strictly follow the instructions, everything will be accurate. The next protocol type is drug testing and animal experimentation. For example, you are testing glibenclamide. Maybe we should have 100 mg per ml or around that concentration. And doses, for example, want to try 2 and 3 mg per kg body weight. Well, it should be per kg body weight, okay? Mg per kg body weight. Administered volume is also per kg body weight. So I will say 1 ml. And then time intervals, for example, we test only one time condition, 1 hour. We can test as many intervals as you wish. Then we use 3 animals per condition, and so we need 12 animals. So we have to enter 12 weights here. Let me say, and another maybe 700. 12 animals, so 12 weights here, maybe a rabbit, there are small rabbits, say grams, and enter. This is the animal protocol, okay? Here, if you mix the solutions exactly in this proportions, you will get a table here where the dose is normalized to per kg body weight, and the volume is also simultaneously normalized to per kg body weight. So this is a critical calculation that the protocol does, and you have to get creative with this. Otherwise, I am not claiming to cover all the scenarios. As you know, animal experimentation is a huge area, but this calculation is critical enough for all scenarios actually. The next protocol type is compound assays, basically analyte assays. For example, if we are assaying the thyroxine in human blood, we have a stock solution of 10 mg per ml, and we can request up to 6 final standards. Free thyroxine in our blood is in the picogram range, picogram per ml range. Okay, let me select picogram per ml. Assay volume 1 ml, and incubation time maybe 12 hours. This is a radioimmunoassay, and then number of samples 12, and we are running the assay in triplicates. Enter. So the volume is very low because of the difference in the stock and final concentrations. Then we have to specify a volume. I will say 10 microliters. That is to be delivered to the assay buffer, and enter. So this is the protocol. These are the instructions. The instructions are in the reverse order, but you have to arrange the tubes in the proper order before adding in order to avoid the confusion. Next, I added a few conversions of units that might come in handy in biochemistry. In this case, this is convert, and we have to select an item from the drop down menu and when we select, the units are also determined at the time of selection, and we need to enter only the numbers. So I will first do all the conversion, show you, demonstrate them, and then explain them to you. Let me go to the first one, mg per ml to millimolar. Here we have to enter the milligram, mg per ml. I will enter only the number, for example 100. Okay, and we need to provide the molecular weight in order to convert the mg per ml units to molar units. Say molecular weight 333 whatever, and okay, so 100 mg per ml is equal to 300.3 millimolar and 10% concentration units, and that is for a molecular weight of 333. Next, the reverse, that is millimolar to mg per ml, 100 millimolar to mg per ml, say molecular weight 222, okay, and this is the conversion. Next, weight to volume. If we have a liquid and we want a certain weight of that liquid, but we don't want to weigh out, instead we want to measure the volume. So say for example 100 grams of some liquid that we want, okay, and then we have to enter the specific gravity. For water, the specific gravity is 1. For other liquids, it can be either higher or lower. Let me say we have a liquid of specific gravity 0.95 and say okay, then the weight equivalent, the volume equivalent to 100 grams of that liquid is 105.3 milliliters. Next, RCF to RPM. If we want the G-force at a particular value, let me enter say 8000 G-force we want. This is important for publications where we report not RPM but the RCF, relative centrifugal force, instead of number of revolutions per minute. So enter. Then we have to enter the radius of the rotor, which we can get from the user's manual of the centrifuge. But here I will put 10 centimeters, it should be in centimeters. It says that here, and then okay. So if we want to achieve 8000 RCF for this particular radius, we have to set the centrifuge speed at 8500 RPM. Next, CPM to DPM. CPM varies from counter to counter. DPM is the standard. So in this case, CPM we can enter multiple values, say 10,000 and 12,000, 30,000, etc. And then that is the CPM. Enter, and then the counting DPM varies with the efficiency of the machine to count. So here normally the power range, let me put it at 40%. 40, in all cases you will enter only the numbers. If you read this, you will understand. If you read this message, okay, and see, this is CPM and these are DPM, and they are also equal to microcuries in each case. And then let me go to the next one, convert optical density to DNA and RNA. Then here say I have a, so if you measure the solution's optical density at 260 nanometers in a quartz cuvette of 1 centimeter path length, then these conversions will be correct. Then let me say we have 0.3, 0.25, 0.6, 0.55 etc. Okay, we say they have to be separated by semicolons in all cases. And then okay, so this is the conversion into actual microgram per ml quantities, units. 0.3 OD optical density at 260 nanometers is equivalent to 15 microgram per ml double-stranded DNA, or 9.9 microgram per ml single-stranded DNA, 12 microgram per ml single-stranded RNA. If you want to find out the concentrations in the actual stock solution, you have to multiply this value with the dilution factor, the times dilution. Next, I will quickly show you how to save the work done here. For example, let me have some items here. First, prepare solution, for example. Name of the compound, glucose. So we have collected some items here. I will show you how to save them. Just go to save. Okay, I am saving it on my G flash drive. Give it a name, save. And you can see here, go back to the file explorer and go to G drive, and let us see where it is saved. See, this is here, testing 2 RST. And if you want to retrieve it, double click on this, and you can retrieve it actually in any of these Microsoft word processors. Let me say we can do it in Word. Okay, yeah, you can see it here in Word format. You can also retrieve it into this program. Let me first erase this. For erasing, Control A, that highlights, and then delete, and then open this one. You can, same work is retrieved here. For printing this, you can click on this print document option, but this is not very sophisticated. I suggest you retrieve it in the Word file, and then actually you can print from there.